Now, this lecture is on stem cell biology and its applications and importance in ophthalmology. To define stem cell, we have a classical definition that has been there for a very long time. And a stem cell is one that has two properties. The first one is capability of self-renewal, and the second one is potency. And by self-renewal, we mean capability of cell division, maintaining its undifferentiated state. It's a cell that can divide without differentiation, because in the body there are cells that differentiate when they divide, and there are cells that die without division. So these are self-renewing cells. They remain as they are when they divide. They have the capability of division without changing their undifferentiated state. And they have potency. This potency means they, when they divide, they can besides give specialized cell type. And we will describe later the meaning of pluripotent, multipotent, or unipotent, or progenitor cells. The embryo, as we know, this is a reminder, is formed when a sperm and an ovum unite. With the formation of a zygote, 2-cell zygote, then a 4-cell zygote, then an 8-cell, and a 16-cell, which is the morula. And this morula starts to have a cavity, and it's then changes into a blastocyst. And this blastocyst starts differentiation into the 3 germ layers, the ectoderm, mesoderm, and endoderm. And this ends in the formation of a whole fetus with its systems. So a sperm and an ovum, a blastocyst, finally, subhanallah, blood, neuron, muscles, and everything. And a stem cell is capable of differentiation into a nerve cell, blood cell, liver cell, cornea cell, retina cell, whatever. Do these all originate from the same cell? Yes, this is what we are saying. They all originate from the known early stage. I don't know why the slides are. Okay. Originally 2 cells, and these form all what you know in the human body. The potency might be totipotent, and these are the cells of the morula, the 16 cells. They can form all types of tissues in the body, and then they differentiate into pluripotent, where certain tissues only can occur from, or a unipotent, where they form, for example, myocardioblasts, just one type of cells. So toti, pluri, multi, oligo, or uni. Totipotent means can construct a complete organism. Pluripotent are descendants from the totipotent and can differentiate into nearly all cells, but not all cells. Multipotent into a number of cells. Oligopotent, only few. And unipotent, 1 cell type. So the potency is the second character of stem cells. So we differentiate or classify stem cells according to their potency into toti, pluri, multi, oligo, or uni, and according to their origin into embryonic and adult stem cells. Embryonic stem cells are isolated from the early embryo, from the inner layer of the blastocyst, and these are pluripotent. They are not totipotent, and if injected in another organism, they lead to the formation of a teratoma. And you remember, a teratoma is a tumor, might be benign or malignant, that is formed of the 3 germ layers. As we said, if you remember in the lecture on embryology. Adult stem cells are found in our bodies, in adult tissues. They repair the systems, reform specialized cells, and maintain normal regeneration. And we have many examples of this. The nearest to us are the stem cells in the palisades of Vogt at the limbus of the cornea. Embryonic stem cells are either fetal or amniotic, and the fetal are the primitive cell types found in the fetus. They say that the classification is unclear, but it is a matter of ethics, matters of law, matters of religion. If you take these fetal stem cells, you are jeopardizing a life, you are destroying a whole life. So currently they are classified as adult stem cells with no clear reasons. Those from the amniotic fluid around the fetus are multipotent, active, and expand extensively. So the embryonic stem cells might be fetal or amniotic. These are human embryonic stem cells in a cell culture under the electron microscope again. Adult stem cells have two capabilities: to divide into a similar cell and divide into more differentiated cells as well. Stem cells in the cornea divide all the time, forming stem cells, and at the same time divide, forming keratocytes, differentiating into keratocytes. Pluripotent adult stem cells are rare. Pluripotent are rare, only found in the umbilical cord, and mostly multipotent mesenchymal and endothelial. Adult stem cells have been used in the treatment of leukemia for formation of fresh, non-malignant blood elements, and in animals in formation of tendons after tendon injury in horses that necessitated previously the killing of animals, of the horses. The research on adult stem cells is not as controversial as on embryonic stem cells and does not require destruction of a life, does not require destruction of an embryo. And if you have stem cells proliferated and you form tissue, this can never be rejected as it is an autograft. And there are many, I remind you of the types of grafts, and there are many government funds in the UK and the US. So the stem cell here may divide into a stem cell, and the stem cell may divide into a stem cell or a differentiated progenitor cell. And this progenitor cell can divide into a progenitor cell or into a terminal differentiation of a tissue. Progenitor cell is the one resulting from a multipotent cell before its differentiation. Now, what's the present state of the art? What's the present condition of stem cell biology? They are grown and transformed. The adult stem cells are grown and transformed in cell culture, and those taken from the umbilical cord blood or from the bone marrow are routine practice. What's the potential for the future? The embryonic cell lines and cloning, human development research, and drug development testing on individual cell types. But this is fraught with hazards, fraught with religion and cultural restrictions, because you are managing cloning, you're managing copying of human body, copying of tissues, and all this is well known to sometimes collide with religious rules, whether Islam or Christianity. In 2012, that is almost 10 years now, Shinya Yamana is a Japanese scientist, biologist. He deserved Nobel Prize for reprogramming adult somatic cells into pluripotent cells. That means taking a skin from the basal cell layer of the skin, for example, and taking a cell from the basal cell layer of the skin and reprogramming it into a stem cell that might have a pluripotent capability. And these cells were called induced pluripotent stem cells, IPS, where they can be taken from patients with a set of the genes that resulted in specific disease and replacing them in another individual treating the disease. These are the potentials for treatment: baldness, blindness, deafness, organ malignancy, wound healing, amyotrophic lateral dystrophy, myocardial infarction, diabetes, bone marrow transplantation, central nervous system like Alzheimer and Parkinson's, where all trials of treatment are almost a failure so far, muscular dystrophy, spinal cord injury, Crohn's disease, all these are potentials for stem cell therapy. But there's a big controversy, as I said, surrounding the embryonic stem cell research. The opponents, those against this research, say this slopes into reproductive cloning, that is copying of human beings in the laboratory, and this devalues human life. This is the thinking of opponents. Supporters say that it is having a significant medical potential and can be done on extra or excess embryos in vitro fertilization. In vitro fertilization, IVF, أطفال الأنابيب, usually they form small embryos that most of them die, and one may continue in successful trials, and these might be the substrate or the platform for this research, but we need all the time both legal and religious approval. I am reminding you here that a graft taken from the body to the same body is an autograft, taken from an identical twin is an isograft, taken from another human body is an allograft, and taken from an animal is a xenograft. Autograft, isograft, allograft, or xenograft. Now we come to our area of limbal stem cells, where the limbal palisades of Vogt contain the corneal stem cells, and these are the actual barrier to conjunctival epithelial cells. Here, as you can see, this is the basement membrane at the palisade of Vogt, where the stem cells occur. These are the stem cells, the red ones. Now, this is the cornea and the limbus and the conjunctiva. These are the palisades of Vogt. Here they are, and here they are where you can see the stem cells. There is a specific disease entity called limbal stem cell deficiency, where the stem cells are deficient, and here we know their value. There occurs then conjunctivalization of the cornea, and there starts vascularization at the periphery of the cornea with global cell instability and irregular epithelium. And this leads to opacification of the cornea, vascularization, chronic dry eye, and all this is called limbal stem cell deficiency syndrome. There are trials of taking the limbal stem cells and their replication in the laboratory. A 2 to 4 millimeter limbus biopsy taken on a special culture medium, where the cells multiply till they become a 2 centimeter sheet, that's almost tenfold, and then they are transplanted to the diseased one. And there is another potential in human amniotic membrane tissue culture. Amniotic membrane tissue culture is prepared, and the limbal biopsy is harvested, as I said, square millimeters, 1 to 2 square millimeters, and these are cultured on the amniotic membrane tissue. where the limbal tissue is digested with an enzyme, and this causes the release of pure stem cells. These stem cell cultured on amniotic membrane become a big sheet of stem cells that can be used in the treatment. I'm sorry. This treats pterygium with prevention of recurrence and treats limbal stem cell deficiency syndrome. The amniotic membrane, being one containing adult stem cells. By the way, we say embryonic and adult cells, not embryonic meaning belonging to the embryo, and adult belonging to the adult. Embryonic means the cell is embryonic. It is from the morula, or adult cell, that is an adult cell, but again, it's belonging to the embryo, taken from either the amniotic membrane, the amniotic fluid, or the amniotic cord blood. Amniotic membrane, with its potential, enhances wound healing, has an antimicrobial potential, and its common uses are chemical injury and limbal stem cell deficiency. The amniotic membrane is composed of 4 layers: the epithelium inside, the basement membrane of the epithelium, and a compact layer and a fibroblast layer. It is prepared by some companies, and there is an Egyptian drug firm that prepares the sheets of amniotic membrane. They are taken and unlaid on the globe with many indications, but you should always make sure that epithelial side is on the outside. Thank you very much for attending this lecture, and now I am ready for your questions.